Hello students, good evening. So good evening. I welcome you all for the biology detailed solutions of the mock test 4. Good evening students. So today we are going to see the mock test 4 questions and solutions discussion part 1. So I think let us start the session. So we'll go for the question discussion. So the question 1 here is, the plasma resembles in its composition to the filtrate produced by the glomerulus except for the presence of glucose, chloride, amino acids, proteins. The question 1 here is, so here which would be the option? So the question asked here is the plasma. So we all know that what happens in the glomerular filtration. We know that the plasma, okay, it is getting filtered, but it is having the same composition of filtrate. That is why we call that as ultrafiltrate, okay. That is why it is called the ultrafiltrate, but it will not have what are the substances that it doesn't have? It will have the composition same as plasma except what? The glucose, chlorides, amino acids and proteins. Okay, so what should be the answer? Yes, you're all right. Yes, so what happens? It gets filtered. So what will not be there? It will be the proteins and RBCs. So as you all know that the filtration membrane, okay, the filtration membrane, it is lined by we have what is called the glomerular capillary membrane, then we have the basement membrane, and then we have what is called the Bowman's capsule epithelial layer, which we call it as podocytes. So what happens? This basement membrane is basically negatively charged, so it will not allow the proteins or the RBC to pass through. See, let us see here. See, so it will not allow the charged particles at large size. So the proteins cannot get filtered nor the RBC. So what happens? You don't usually find it in the glomerular filtrate, as well as we don't find it in the urine. So the option would be proteins. Okay, so that is the right answer. Whereas the glucose, chlorides and amino acids do get filtered, but what happens there? They get filtered, but at the same time what is going to happen there? It is going to be getting reabsorbed by the renal tubules. So that concept you should know. Okay, we'll go to the next question here. The next question here is, which of the following has the maximum water potential? Pure water, 2% sucrose solution, 4% glucose solution, 10% sodium chloride solution. So first we should know what is water potential. I hope you all know this pi, which is denoting the water potential. It is the potential energy of water, that is per unit volume, that is corresponding to the pure water, which is based on the mentioned circumstance, that is they are expressed in units of pressure. So when water potential is decreased with increase in solute concentration, so that you should know. So pure water has got the highest or the maximum water potential, because which is not under any pressure and it is at the standard temperature when it is taken at 0. So the osmotic potential of pure water, that is when you see that maximum water potential, okay, here it will be usually what? It will have the maximum water potential. So you can see the water potential, which I already explained. It is nothing but the difference in the free energy or that chemical potential, whichever is there per unit molar volume of water in the system, and that of the pure water at the same temperature and pressure. So that water potential of pure water is 0, because I already explained that it decreases on the addition of solutes. So the answer would be pure water. Yes, good evening students, good evening. So please do participate in the question and solution for the biology session. Introduction of food plants developed by genetic engineering is not desirable, okay, because options given are: economy of developing countries may suffer, these products are less tasty as compared to the already existing products, the method is costly, transgenic food may cause toxicity and allergy in human beings, and the bacteria present in the alimentary canal may become resistant to antibiotics by taking up the antibiotic resistant gene that is present in the genetically modified food. So when I think about this, see, genetic engineering is made, probably what they want to improve the quality as well as the quantity also of the food plants, because they want to meet the demands of the growing, the exploding population. But how is this being done? So what is that you need to know? Like when you see the economy of developing, so when you look into most of the thing, they say that the economy of the developing countries may suffer, but this is not the actual solution. When I went through and reviewed some of the research article, they are saying that the transgenic food, no, it is causing the toxicity, that is particularly allergy in the human beings. So what is happening? The entire lining of our alimentary canal, okay, it is becoming resistant to that antibiotic, because when we are taking some genetically modified food, that antibiotic resistant gene is getting incorporated, and that is bringing these changes. So that is one of the drawback. So the biotechnology in future advancement, they have to take steps in overcoming what? In overcoming these sort of complaints in terms of allergy and toxicity. Okay, but when they say in the contrary, the next real option is the economy. So developing countries, when you see, they cannot afford to meet with the sort of producing such genetically modified plants also. So they say the economy of the developing countries may suffer. But here the right option would be this. Okay. So the transgenic food may cause toxicity and allergy in human beings. Okay. Yes, please do post your answers in the chat, children. An institution where valuable plant material likely to become irretrievably lost in the wild or cultivation is preserved in a viable condition is known as genome, herbarium, gene library, gene bank. So when you look into this concept, okay, so we should know that those valuable plant materials, where are we going to keep them safe? So as we say, like how valuables we do keep safe in the banks, lockers, same way we have what are called the gene banks. These are a type of repository where the genetic material can be stored. Okay, so like same way in humans, like even in many of those endangered species, something like where we can keep this in vitro storage, freezing, cuttings from the plant, stockings, the seeds like in seed bank. So even for animals, you know, the freezing of sperms and eggs in zoological freezers, they are kept until further need. So these gene banks, okay, what they do here is that plant genetic material, whatever, they are going to preserve at minus 196 degrees centigrade in liquid nitrogen, and as the mature seed or whatever. So once when it is need, they'll unfreeze it and then they use for the propagation purpose. So this is how they do. With corals and all, no, they take those fragments, okay, and they'll be storing it in the water tanks under those controlled conditions. So this is how they do maintain, like keeping something which they need to preserve it, because they are endangered or they might be threatened, like that. Okay, so it is the answer, the right answer would be gene bank. Yes, yes students. The certain species of wasp are seen to frequently visit, okay, the flowering fig trees. The interaction between them, okay, the interaction between them is called the brood parasitism. or it is called the mutualism, or it is called commensalism, or it is parasitism. So what should be the answer? So when I say between a wasp and the flowering fig tree, brood parasitism is different. That is between that crow and cuckoo, right? Commensalism is different. Yes, like neither benefit nor parasitism. One is getting benefited, the other is harm. But here that visit is going to benefit both. So what we call that as mutualism. So mutualism is a way where two organisms of the different species, where they exist in a relationship, where each individual are going to get benefited. Like how we say that cooperation, what we call. So the association between that mycorrhizae, okay, and that symbiotic relation, what we call. Okay, so because they get in terms with that nutritional support, like that are examples of mutualism. So same way here, the female wasp. Okay, so you can see those female wasp, what they do, they will just go and they will be laying those eggs. Okay, that we not only as oviposition, but they use that developing seeds within the fruits for nourishing their larva. In turn, the wasp, okay, help in pollinating the plant. So like I am helping in pollinating, and in turn you are going to feed the larva, whatever my young one. So there is a mutual benefit of both the organisms here. So the fig offers the wasp some of its developing seeds also. Like you are helping in pollination and I am giving the payment for it. Like that, it's in a form of reward. So the best example here would be the mutualism. So what we see here. Okay, it's not with other. So this is one of the best example we can say. Okay. Okay, here we go for another nice interesting question, which we say in terms with identify the incorrect statement from the following, okay, with reference to lac operon. Okay, so the options given here are, it's a unit of gene expression and regulation for lactose sugar metabolism in E. coli. Lactose sugar enters the cell due to the activity of the enzyme permease. Operators are present between promoters and structural genes. The structural gene is at code for beta-galactidose and Y for trans-acetylase and A for permease. So we have to find out which is the incorrect statement. Let me tell you that lac, okay, that lac operon is an operon, okay, it's involved in the regulation of those lactose metabolism in several enteric bacteria, particularly even the E. coli also. So it has got three units, okay, it will have a promoter, it will have what is called the promoter, it will have what is called the operator, okay, and it will have what is called those three structural genes and a terminator. Okay, so these will be there and they will have a terminator. So this is the real what you call the structure of those inducible operon. So there are three structural genes, okay, there we have what is called the LAC-Z, we have LAC-Z, then we have that Y and we have A. The Z will always code for the beta-galactidose and the Y will code for the beta-galactidose permease and A will code for that beta-galactidose trans-acetylase. Okay, so this beta-galactidose, that what it will do, it will cleave the galactose into glucose and galactose. The permease, that will help in transport of the lactose sugar into the cell. The trans-acetylase will help in transfer of that acetyl, that is acetyl-CoA to that beta-galactose. So that will be the role actually. But what is happening? See, it is a unit of gene expression and regulation for lactose sugar metabolism in E. coli is correct. Lactose sugar enters because of how? Because of the enzyme permease. The operators are present between promoters and structural gene. But what is wrong here? The Z. So what is that? Z is for what? The galactosidase. What about the Y? It is for the permease. And what about the A? It is for the transition. So this is wrongly given. So that is the odd one out. So you are able to follow. So that is the odd one out here in the that is the incorrect statement. Yes, yes, very good, Nilambari. That is the one. Okay, a sugary solution is changed to vinegar by the action of, okay, acetobacter. Okay, it is changed by acetobacter. or Acetobacter aceti, Bacillus subtilis, Mycoderma. So what should be the right answer? So we should know that what happens during the process of production of vinegar, the sugar will get fermented. Okay, the sugar is the one which will get fermented to alcohol, and that alcohol is again further fermented to this acetic acid, probably by the use of this bacteria called Acetobacter aceti. So that is the one which is used for the preparation of the vinegar. So during the production of vinegar, the sugar is the first fermented to alcohol, and see, the alcohol is fermented further to acetic acid. So Acetobacter aceti, that is the one is used for the preparation of vinegar from the fermented sugar solution. So that is the right answer. Okay, which one engulfs pathogens rapidly, whether it is the acidophils, erythrocytes, basophils, neutrophils? So what should be the right answer? See, let me see what are the options. See, now see, acidophils, they are a group of cells which you find in the pituitary gland. Okay, the pituitary, anterior pituitary gland histology, when you take, you have a group of cells, what you call them as acidophils, and what you have what are called the basophils there. So those acidophils group of cells have a characteristic from where they secrete the growth hormone and prolactin. So this will not be involved into it. What about the erythrocytes? The erythrocytes are the RBC, which is going to help in the gas exchange, and they are going to be the good buffers. What about the basophils? The basophils, along with the mast cell, they are going to play a very important role in hypersensitivity reactions. Okay, so which should be the right answer here? Okay, so the right answer is, excellent students, you are giving the right answer. It is the neutrophils. So the neutrophils, which we call it as polymorphonuclear leukocytes, polymorphonuclear leukocytes. Okay, the other name for neutrophils. Okay, when you look into the neutrophil, okay, they will be like this. So they will have many nucleus, and they will have those granules also. Okay, so you can see the granules there, right? So these granules will be there. So this is the structure of the neutrophil. So these neutrophils are white blood cells. They are going to protect our body from various pathogens, and probably how do they do? By the process called phagocytosis, which means I engulf. Which means I engulf. Okay, that means I eat. So what they do? So they, suppose, imagine this is the bacteria. What will happen? They just go engulf in it, and they will have that powerful lysozymes in that, and that is how they form that phagolysosome, and that will end in the process of phagocytosis. So neutrophils engulf the pathogens in our bodies. So they are special type of WBCs where they protect our body from various pathogens. So the answer would be neutrophils. Okay, the answer would be neutrophils. So we'll see the first event in photosynthesis. The first event in photosynthesis is synthesis of ATP, photoexcitation of chlorophyll and ejection of electron, photolysis of water, release of oxygen. So when I say the first step in photosynthesis, what should immediately strike your mind? So immediately it is the photoexcitation. So what is happening? It is the photoexcitation. Okay, that is the main, the pilot event that is taking place in the photosynthetic process. So what happens when those light photons, when they fall on the chlorophyll molecule, the electrons which is there in the ground state get excited up. Okay, so there is a passing from the, what you call, from that base state into a higher energy state, what we call the signet state. Okay, so you can see here what is happening here. Okay, so once when the light molecule falls on it, so what happens when the light falls on it? They get excited up. Okay, so what is there happening? There will be passing of the energy level. So from the lower energy, it just passes up to the higher energy level, which we call it as the signet state. Okay, so this happens here. So photoexcitation of the chlorophyll molecule and ejection of electrons. So that is very important. So you have all these steps. ATP is going to get synthesized. There will be photolysis of water. and release of oxygen, all that is fine. But the first event, the pilot event, that is the photoexcitation of chlorophyll and ejection of electron. Now we will see, choose the correct statement from the following. So we have to find the correct statement. Yes, very good students, you're participating very well, very good. So the dioecious, which we mean the hermaphrodites organisms, are seen only in animals. Dioecious organisms are seen only in plants. Dioecious organisms are seen in both plants and animals. Dioecious organisms are seen only in vertebrates. First we will see what is dioecious. That means having male and female reproductive structures in a same, like you don't find it separate organism. So having the male and the female reproductive organs in separate individuals, they are called the dioecious. So dioecious organisms are seen in both plants like papaya and animals like cockroaches also. So dioecious, they are called the hermaphrodites. So what is the common thing that we see? So the dioecious organisms we see in both plants and animals. Okay. So antibodies are synthesized by eosinophil, lymphocyte, monocyte, neutrophil. So what should be the right answer? See, antibodies are otherwise called as immunoglobulins. Okay, they are going to protect our body from various pathogens. So antibodies are actually produced by B lymphocytes. See, lymphocytes are actually classified into T lymphocytes and B lymphocytes. The B lymphocytes, okay, they will produce the plasma cells. They are the precursors, and from that only the antibodies are produced. So immunoglobulins, you know, there are 5 types, okay, IgA, IgE, IgD, IgM, okay. So these are the immunoglobulins, and IgG. So these immunoglobulins are the antibodies, okay. So they are synthesized by the B lymphocytes. So what should be your right answer, ma? It is not the eosinophil, because eosinophil are with anti-allergic and anti-parasitic effect. So their role is something different. Okay. What about monocyte? Phagocytosis. They are the second line of body defense. Even the neutrophil, the first line of defense. Phagocytosis. So what should be your answer? It is the lymphocyte. So see the cells beautifully. So these lymphocytes, okay, they will be somewhere around 7 to 10 micrometer in diameter. Okay, so you have T lymphocytes and B lymphocytes, and they are the one which is going to produce the antibodies, particularly the B lymphocyte. Okay, so that you have to narrow it down. Yes, lymphocyte is the answer. Myelinated nerve fibers are white colored because of chromosomal substance, neurolema, myelin, none of these. So when you look into the structure of the nerve, okay, particularly these axon, which we call the nerve fiber, acha? So these are the dendrites, okay, and then we have that covering there. It has got a very beautiful covering which wraps up this entire of the axon, which we call it as the myelin sheath. So this gives that characteristic white color. If they are myelinated, it will give that characteristic white color. So the myelin sheath, okay, that appears as tube around the axon. It will be like on plastic, like it just wraps around. Okay, it is a mixture of lipids and proteins which we call the myelin. Particularly the myelinated sheaths will be white in color. Okay, so that we need to know. The neurolema is that outer covering, and chromatin substance that will not fit into it. So the answer is myelin. Yes, we'll go to the next question. The capacity of an environment to pull on a limited number of individuals is known as bearing capacity, limited capacity, environmental resistance, and carrying capacity. So when I say carrying capacity, they are talking in terms with size, the size of a population that can be maintained indefinitely. So they are talking about the maximum number of individuals of a given species, the area or resources that can sustain indefinitely. Okay, so carrying capacity refers to the size of population that can be maintained indefinitely, and carrying capacity is the maximum number of individuals of a given species that an area resource can sustain indefinitely without significantly depleting or degrading those resources. So they are able to take it in that. So we call that with the capacity of that environment to take that or pull on to that limited number of individuals, we call that as the carrying capacity. Yes, very good students. Next we will see about what is anabolism. Okay, anabolism is an endogonic process, exogonic process, bidirectional process or destructive process. So when I say anabolic process, it's an endogonic reaction in which the products, okay, has more energy than the reactants as they require an input of energy. Like say for example, like all the small amino acids, okay, they just get built up, which is an anabolic process where a protein molecule is formed. So that is how a growth of a cell or a tissue is being brought about. So it's anabolic. Okay, so when I say anabolic, what happens there? It's a biosynthesis or endogonic reaction in which the product has more energy than reactants, so they require an input of energy. So large molecules, when they break down, they go for small molecules, which we say catabolism. When all the small molecules, I said no, take tiny glucose molecules, when they construct together, they form that glycogen, okay, a large molecule. So that's an anabolic effect. Okay, whereas the glycogen in turn is breaking down to small glucose molecule, catabolic effect. So that is catabolism. So the answer would be endogonic process. Yes, very good students. The seminal plasma in human males is rich in glucose and calcium, DNA and testosterone, ribose and potassium, fructose and calcium. So the seminal plasma, so it is rich in what? So when I talk about the seminal fluid, the semen, which will be ejaculated during the sexual intercourse, nearly around some millions of spermatozoa will be released. So the semen is actually a combination of fluid released from the prostate gland, the seminal vesicles and the bulbourethral glands. Okay, so it not only gives the medium for it to transport, it is going to nourish the sperm, it is going to help in lubrication during the process of sexual intercourse, and it is going to neutralize that acidity that is there in the vagina of the females. So it has got a rich source of fructose and calcium. So you see the seminal plasma, it is secretes around 60% of the seminal plasma. It is more alkaline. Okay, so mainly to contract that pH, which I already said. So it has got a main nutrient which is called the fructose and other mucus and other coagulating enzymes. Prostate gland secretion for 30%, which will have that citrate, and it has got that nutrient for that sperm mitochondria, and calcium and various proteolytic enzyme is there, that is termed as prostate specific antigen. That is why in men after that 40 years or 45 years, if they come to us with complaints of they are having in difficulty in passing urine and all that, we will ask them to check for this particular antigen, which we say prostate specific antigen, if we are suspecting, and we can take it through the blood test and we will verify whether they are having any hypertrophy or carcinoma like that. Okay, so that to liquefy and ejaculate just before the release. And that Cowper gland, that bulbourethral, will add for small volume, which meant for lubrication purpose. Okay. What is correct about the mechanism of hormone action in humans? So when you talk about the hormone action in humans, glucagon, you are going to see which is correct. Glucagon is secreted by the beta cells of islets of Langerhans and stimulates glycogenolysis. Secretion of thymosin is stimulated with aging. In females, FSH first binds with specific receptors on follicular cell membrane. FSH stimulates the secretion of estrogen and progesterone. First we will see about glucagon. Glucagon is secreted by alpha cells. Nowadays we don't use the word alpha or beta, we call it as A cell and B cell. So A cells of that will secrete the islets of Langerhans, they secrete the glucagon, and they are not meant with glycogenolysis, rather they are the one which is going to stimulate what is called the deposition of glycogen. Okay, they are not going to help in breakdown of they are going to stimulate the glycogenolysis, because insulin is the one which is going to help in converting the glucose to glycogen. But glucagon is something in terms along with the need, it will come out immediately whenever the blood glucose level goes down. The glucagon has to come along with other hormones to increase the blood glucose level. So the glycogen will be broken down to glucose here. So this part is correct, but what about here? This is wrong. So this is not the correct statement. Secretion of thymosin. Thymosin is a hormone which is secreted by the thymus gland. Okay, so that is needed for the particularly the development of T lymphocyte. Okay, what we call it as pre-processing. So and you have to know that it is something related with immunity. So this will not be the option. In females, first you should understand that the FSH will bind with the specific receptors on the follicular cell membrane. Yes, this will be the right answer. Okay, so see, glucagon is secreted by alpha cells of islets of Langerhans and stimulates glycogenolysis, breakdown of glycogen into glucose. Thymosin comes from the thymus gland, stimulates the development of various kinds of WBC. Children, be careful, it is only the T lymphocyte, and it also hosts the attainment of sexual maturity. What about in female? The FSH will bind with the follicle, that is needed for the follicular development, puberty, maturation and other things. FSH will stimulate mainly the follicular development. So it is not with the secretion of both estrogen and progesterone. Okay, progesterone comes from the corpus luteum. It is actually the LH will stimulate both the secretion of estrogen and progesterone. Okay, yes, very good, you have given the right answer. Which one of the following is not a plant like protist? Desmid, dinoflagellate, diatom, slime mold. So when you see the what you called the concept, when you see the desmid, dinoflagellate, diatom, they are all plant like protist. Which is the option? The fungi like protist is the slime mold. So see, the slime mold is a fungi like protist. Desmid also they have a clue. Many students know, you know that dinoflagellate, diatom, they are all direct. But what about them? You will have confusion between the desmid and the slime mold. Desmid also, they are also single cell green algae. Okay, and because of their internal chemistry, now they have come up and attained a common place in the kingdom Protista, like with other forms of microalgae. So this is also not, only the slime mold is not a plant like protist, where they are included as fungi like protist. Okay, so that is the right answer. So see here, the desmids which I told, the dinoflagellate, diatom, they all are plant like protist. This is only the fungi like protist. Okay. In order to obtain virus free plants through tissue culture, the best method is embryo rescue, anther culture, meristem culture and protoplast culture. So we have been doing in sessions together. The right answer is the meristem culture, because we know that is a place where there is development of virus free cells, because every time there is a renewal there, because it's a growing region. The meristematic tissue, you can take it either from the shoot tip or the root tip. So the exact answer is the meristem culture. You have to take it from which place? It is the virus free from the meristem place. The characteristic feature that can be exclusively seen in angiosperm is seed, fruits, endosperm, syngamy. So what is so characteristic that you see about angiosperm? The angiosperms are called the phanerogams, right? So that is characterized by what? The flowers as the reproductive organs, because of the presence of other structures like root, stem, leaves. The characteristic is the presence of those flowers. Okay, so the fruit formation is the characteristic feature of angiosperm. Like you have no fruit formation in gymnosperms, like because there is no ovary. So the seeds which are present in gymnosperms as well as, but they do not have flowers. So the seeds are said to be naked, right? So in angiosperms, the seeds are covered due to the presence of ovary within the flower that ripens and later that becomes fruits. So gymnosperms do not make fruits. So what is so exclusive that you see about angiosperm is the fruit. Okay. Eustachian canal connects middle ear with external ear, middle ear with internal ear, middle ear with external ear with internal ear, middle ear with pharynx. So when you talk about the Eustachian canal, okay, you can see here the middle ear. The middle ear comprises of those three bones we call the malleus, incus and stapes. We have the tympanic membrane, the eardrum, and then we find what is called the tube here, which we call the Eustachian tube, or it is also called as the auditory tube also. So this is the one which is going to connect the ear with the pharynx. Okay, so the middle ear is connected with pharynx by the Eustachian tube. So that is going to bring about a balance between the outer atmospheric pressure and that with the body. Like particularly you can experience that ear block and all, like when you travel up in the airplanes or something, like when you yawn up, you have that relief and all that. So probably that is because of this block in that thing which we call the eustachian tube, the auditory tube that is going to connect the middle ear with the pharynx. Yes, very good children, you are giving the right answer. Now we will see the next question. The ratio between the 2 carbon and the 3 carbon intermediates having the NH2 group formed in photosynthetic oxidation cycle is 1:1, 2:1, 3:2 and 3:4. So when it comes to the photorespiration part in peroxisomes, particularly where it takes place, the molecules of glycine, what happens, that is 2H2N CH2 CO2, they are transferred into the mitochondria, where they will be converted to one molecule of that serine. So what is happening? So that ratio of 2C and 3C with that NH2 will become in the ratio of 2:1. So that is the right answer. Some common marine fishes are hilsa, mackerel, pomfrets, all of these. So we are very well aware of all these three. They are the what? They are the common marine fishes. Like you can see the hilsa, okay, the Indian shed we call, and then we have those mackerel, and then the sardine, the pomfrets. So they are all the common marine fishes. That is Sardinella longiceps, Harpodon heparis, Bombay duck, hilsa, Stomatiris sinensis, pomfret, Angula L, and Olithronema salmon, and flatfish which is a Cyanoglossus semifacetus. All these are common marine fishes. So hilsa, mackerel, pomfrets, all are what? They are all common marine fishes. Smooth muscles are involuntary, fusiform, non-striated, voluntary, multinucleate, cylindrical, involuntary, cylindrical, striated, voluntary, spindle-shaped, uninucleate. So first you should know that there are three classes of muscles: skeletal muscle, smooth muscle, cardiac muscle. The characteristic here, skeletal is voluntary, these two are what? Involuntary. Skeletal is what? It is striated, cardiac is striated. But what about the smooth muscle? The smooth muscle will be non-striated. So that is another important difference that you need to know. What do you mean by involuntary? They are not under our control. You can't say stomach, come on, do this action. Whereas you can command your fingers, you can command your legs, but you cannot command any of your internal organ, that because they are lined by smooth muscle. So the appropriate answer would be they are involuntary, accepted. They are non-striated, accepted. They will be fusiform shape, they will be fusiform. So that is where, because these smooth muscles, they are involuntary, fusiform, non-striated. They are going to present in the hollow organs like urinary bladder, or they will be present in the stomach, okay, or intestine, in any passage, even in the blood walls, arteries, veins of the circulatory system, respiratory, urinary, reproductive, everything, they are lined up with the smooth muscle. So that is why they are involuntary, fusiform and non-striated. Okay. Yes, very good students. Enzymes that catalyze the transfer of molecules except H and O and electron are called as ligases, isomerases, lyases and transferases. So this is a question which we have said. What are ligases? It's something that is helping in joining of two molecules. Say for example, they are going to break down, forming new bonds with simultaneous breakdown of ATP or pyruvate carboxylase, like that. Ligase is joining two molecules. What about isomerases? Isomerases are related with some molecular rearrangement, like in form of phosphoenamol isomerase or glucose fructose with that isomerases, change of substrate in related form of intramolecular rearrangement, that is with isomerases. When I say lyases, they are going to split that other similar bonds like decarboxylase, aldolase, like that. But what about transferase? Transferase are transfer of one functional group in one substance, say kinase, phosphorylase. So transferases are enzymes that catalyze the transfer, of group of atoms such as amine, carboxyl carbonyl, methyl, acyl, glycosyl, phosphoryl from donor to acceptor compound. So there is a transfer. So that transfer of a functional group from one substance, like example phosphorylase, kinase, etc. So that we can know. So the answer is what? Transferase. Yes, very good students. Prabhanti Kanti is a new variety of Abenolocus esculentus having the resistance to shoot and fruit borer, bacterial blight, yellow mosaic virus, tobacco mosaic virus. So Prabhanti Kanti, when you say, it's a yellow vein mosaic resistant variety which was released in 1985, okay, by Mao, that Prabhani. It is derived from that back cross of that Abenolocus manihot into that Abenolocus esculentus. So two varieties have been back crossed, and with those two back cross we got a new variety which is the Pusa savani, okay, which is Abenolocus esculentus as a recurrent parent, followed by selection in subsequent generation. So this is quite resistant to that yellow vein mosaic virus, okay, variety isolated in that F8 generation. So many generations it have been checked to. So the plant flowers in 45 to 60 days. The fruits will be dark green in color, that lady's finger, smooth, and it will be very slender. So that is the advantage. So Prabhani Kranti is what? It's a new variety, but it is between the Abaculus manihot and Abaculus esculentus. So it is resistant to yellow mosaic virus. Now here you see, here they have given one mRNA segment. Acha? So study the mRNA segment. So we are studying what is given here. This is how when a picture is given, you have to analyze the picture. So something A is there, there they have given all these things. Okay. Now we are going to study the mRNA segment, which is to be completely translated into a polypeptide chain. Now they are asking the codon for A and B. What are the codons for A and B? So here they have given all these. So what are the codon? So when I say codon, the start codon is the first codon of the mRNA. So here, so here it is the start codon. So that is the first codon of the mRNA, the transcript translated to a ribosome. Okay. Now the start codon will code for that methionine. So here the usual start codon will be of what? AUG. Now the B here, it is the stop codon. So when you take the segment, the first will be a start codon, the last will be what? The terminator or the stop codon. So here this is the stop codon. So it is always the triplet, that nucleotide triplet which will be there, okay, that within the mRNA that signals the termination of the translation. So the stop codon, they do not have amino acids. So the common termination codons are what? UAG, UAA and UGA. So when you are going to check in this will not be the option and this will not be the option. So AUG both are there. But what we have to look through here? We have to look for the stop codon. So what is the nearest stop codon? They have given this UAG. So what is the right answer? It is this. Okay, so this is how you are going to make it up. Okay. So the next one, the result of light reaction are only ATP, only NADPH2, ATP and NADPH2, only FAD. So when you take during the light reaction, what happens? The sunlight, those photons are just taken up by the chlorophyll. So here they will be converted to the chemical energy, that is they are stored in the form of ATP and NADPH2. Later what is happening? That will be used during the dark reaction in the night time. So during light reaction, the energy from sunlight is absorbed and they are converted to the chemical energy, which is stored as the ATP and NADPH2. So later they will be used for the dark reaction for synthesis of those organic food products. So an extra chromosomal self-replicating part of the cell that has proven to be a boon to biotechnology is virus, mitochondria, nucleus and plasmid. So when you see plasmids, they are the extra chromosomal self-replicating structures. When you see, so you can see those plasmids, they are extra chromosomal, okay, they are circular double-stranded DNA, they are found in many bacteria, they are self-replicating. So scientists have taken this advantage of these plasmids. They are using as a tool for transfer, for cloning and manipulation of gene. The plasmids are used. So these plasmids, they are experimentally, they are used these purpose where we call them as those vectors. Okay. So researchers can insert that particular DNA fragment, okay, or the gene into a plasmid vector, which we call it as that recombinant plasmid, and then that plasmid will be introduced into the bacterium by a process called transformation, and then the bacteria will divide and they will produce n number of copies of those DNA fragments. So that is how they have done. So the real boon to our biotechnology is what? It is the plasmid. Okay. So the theory of natural selection that explains the appearance of new forms of life on earth was given by Oparin and Haldane. So their experiment was different. So Hardy-Weinberg equilibrium is different. Mendel, it's with different. So Darwin. So here it's the Darwin. So Darwin, there are actually two co-discoverers, they'll say. It's Charles Darwin and Alfred Wallace. So those two pioneers, okay, in 1958 as well as in 59, they put forth the theory of evolution. But who is the pioneer? It is the Charles Darwin. So he was the one who gave explanation for adaptation and formation of new species. Okay, he defined that natural selection, gave the thing of the survival of the fittest. So his topic is on that origin of species, no? So he defined the natural selection as the principle by which each useful variation is preserved. So the answer is Darwin. So anxiety and eating spicy food together in an otherwise normal human may lead to indigestion, jaundice, diarrhea, vomiting. So what should be your answer? Jaundice is yellowish discoloration of skin and mucous membrane due to more increase in the presence of bilirubin. Diarrhea, it can be because of some infectious condition associated with this vomiting. But what about indigestion? Indigestion is regarding when you are very anxious or taking more of those spicy foods and all, no? It will not have that proper digestion, so they get that what is called that reflux of those contents. So that we call it as indigestion. So you can see the indigestion. So anxiety and eating that in a normal healthy man can lead to indigestion because there is difficulty in digesting the food properly. So that could be the reason. Sometimes they might be mistaken in a case with very elderly people, like what we call, we have those differential diagnosis, we say whether it is a real anginal pain, the chest pain, or indigestion, we have to rule out. So that we have to make it up. So indigestion is the answer. Yes students, very good, you are giving the right answer. The action of vaginal diaphragm is to prevent the ova to come in uterus, to prevent the sperm to come in contact with ova, spermicidal, anti-implantational. So when you see the vaginal diaphragms, like okay, there are like what you saw, the contraception, it's one of the contraceptive method which is going to prevent the pregnancy or conception. So like other barrier methods, okay, like how in men we have condoms, like females they use what is called those vaginal diaphragms. So they say the vaginal diaphragm role is to prevent the ova from getting get in contact with the sperm. So that should be the right answer. So you can see that diaphragm, so they will be just inserting it into the vagina. So cervical caps, walls, like dome shaped rubber plastic covers or barriers, which I already said, they are inserted into the female reproductive tract to cover the cervix during the coitus. So they help prevent the conception, so that the sperm, like blocking the entry of sperm through the cervix. So but they say that this method is not that great effective, because they say that you have to use along with the spermicidal jellies, then only it will be a successful contraceptive measure. So regarding that, that's why along with the spermicidal, it's a successive method. Okay. So coming to the next question, a colorblind girl is rare because she will be born only when a father and maternal grandfather were colorblind, a father and maternal grandfather were colorblind, a mother is colorblind, father is normal vision, parents have normal vision but grandparents were colorblind. So when will a girl child get that colorblind? So it's quite simple, that color blindness, when you see, no, it's a X-linked recessive genitive disorder. It's a X-linked genitive disorder. That means the disorder is linked with the X chromosome. Okay, so in boys they have only one X, so it's a single defective gene, and that is the reason that boys will have the color blindness. But in a girl, she'll have two copies of X, one from the father, one from the mother, so that she'll have the two copies, which is quite rare. So if her father has a normal vision, that she will never show the disorder. Hence, what will be the real option? Here you can see that the genotype of the mother. Okay, so here you can see the genotype of the mother was to be either like colorblind or like this, that is XC, or it can be one X defect, another normal, or with the father having this, so that the daughter becomes colorblind. So here the father is colorblind and a mother is a carrier of the disorder. It can be like that. So that the maternal grandfather was colorblind. So the maternal grandfather would have been colorblind. So the mother was a passive carrier. Okay, and her father is also colorblind. Then she has all the possibility of developing it into a colorblind. So that should be the answer. Okay. Bacillus thuringiens, okay, is a good biofertilizer, biopesticide, biofuel and single cell protein. So when you say Bacillus thuringiens, okay, it's a naturally gram-positive soil bacterium which has been from 1950s, no, we are using as a natural insect control. Now it will have that spore that gives a resistance where it produces those protein crystals which is quite toxic. It's not for all the insects, particular to that insect. So once when it is taken inside, those crystals, you know, they become that toxin, and because of that alkaline pH, you know, in the gut of that insect, it is going to kill the particular insect. So what we have to know, the Bacillus thuringiens is a gram-positive soil bacterium that is commonly used as a biopesticide. So that bacteria is producing that toxin, proteaceous toxin, which we call the thurioside, which is insecticidal in nature. The bacterial spores will be sprayed on those insects, pests, flies, moth, mosquitoes, etc. So but they are harmless to those birds, fishes and mammals whose acidic gut conditions will negate this bacteria effect. So specific Bt toxin was isolated from this thuringiens and it is incorporated into several crop plants, cotton, brassica family, like that. So they want to have a good control, particularly it's a very good biopesticide. Okay. Yes, very good children. You are giving the right answers. Good. The reason for the population explosion in the world is increase in the birth rate, decrease in the death rate, both A and B, none of them. So increase in birth rate is that natality, right? What about decrease? The death rate, we call it as mortality. So when there is increase in the birth, decrease in the death, yes, definitely there is going to be explosion in the population rate. So the factors which favor the population is increase in the natality, the birth rate, or decrease in the death rate, the mortality. Like and moreover, there is more people moving from one place to another place, like there is immigration, and there is, because of this increase in their lifespan and with better medical facilities what is available today. So that is the reason for the population explosion, like good care in the terms of neonatal and maternal services. So all these are increasing the birth rate also. So all this is leading to the population explosion. Yes, yes, very good. That is the right answer. Which one of the following cell organelles are enclosed by single membrane? Mitochondria, chloroplasts, lysosomes and nuclei. So when you say single bound membrane, no, remember they are the vacuoles, lysosomes, okay, the Golgi apparatus, the endoplasmic reticulum. They are all single membrane bound organelles that is present only in the eukaryotic cell. So you can see the lysosomes, others are all double bound. So the lysosomes are the one which is having the single membrane. The lysosomes we know, which has got those powerful hydrolytic enzymes, you know, they just pour on the substance which comes in contact with them, even the own cell, or it can be even a pathogen or anything. So it is so what we commonly call as suicidal bags. So that has got so much of powerful enzymes in it. Which of the following cell is round and biconcave in shape? WBCs, RBCs, epithelial cells and nerve cells. So WBCs, I said no, they don't have that constancy in shape, like they are mostly circular. Okay, and what about those epithelial cells? Depending on the area, like it will be either a squamous cell or a columnar or cuboidal. Nerve cells, we know that it has got a perichorion with dendrites and axon. So what should be the answer? Yes, it's a RBC. Looks like it, right? Round, or it's biconcave in shape. See the biconcave shape. Okay, so the shape of RBC is going to be vary. Like in terms in mammals, like they'll be either circular, biconcave and enucleated disc. This biconcave shape, you know, it is given by two main proteins. We call them as spectrin and chirin. And suppose if there is a loss of these proteins, you know, it will make your RBCs more circular, and it will lead to a characteristic disease which we call it as the congenital hereditary spherocytosis. That will lead to anemia. It will become more spherical, ma. Okay, so that is the answer. So These two proteins are important to maintain this shape. If suppose that shape is not maintained, they become more spherical. Once when they pass through the blood capillaries, what will happen? They just get ruptured, leading to anemia. Okay. So the central part is going to be very thinner. Okay, and the margins you can see, the central part will be thinner than the margin. So that is giving more flexibility, increasing the surface area, etc. So that should be the answer. RBCs. Diabetes insipidus is caused due to the deficiency of aldosterone, ADH, ACTH and TSH. So diabetes insipidus is a characteristic condition due to the deficiency of the hormone, antidiuretic hormone, which results in the characteristic polyuria and polydipsia. So the main role of antidiuretic hormone is to reabsorb the water from the distal convoluted tubule and the collecting ducts. So once when the water is not getting reabsorbed, more water is going to get lost. So there is going to be enormous output of urine, dilute urine. So once when the water is lost, the person goes in for enormous intake of quantity of water, which we say polydipsia. So the amount of water excreted in the urine is controlled by ADH, vasopressin. So when there is hyposecretion of ADH, which results in diabetes insipidus. Okay, where the kidneys cannot concentrate urine, so they pass large amount of dilute urine. Okay, so that's the answer, ADH. Yes students, you are right. The Chipko movement was launched for the protection of forest, livestock, wetlands and grasslands. So Chipko movement was launched because for the protection of forest. So the Chipko movement referred to those rural people, particularly in the Himalayan region where they protest in 1970 and 80s. Okay, those Bishnoi family also, they have that characteristic where they were the pioneers where they had, but this Chipko movement was in that 70-80 in the Himalayan rural areas. Those women protested against that indiscriminate logging and felling of those trees. So this was concerned in April 1973. So the next fight, this was spread along overall. So every places like in Uttar Pradesh and everywhere. So they wanted to make like, they want to protect the forest, they don't want to cut the trees. So that is where with the Chipko movement came into existence. So even now that Bishnoi award they give for every year, the government gives to the people who are actively participating in protecting the nature or actively involving in that environmental concerns. So that is where. So Chipko movement is particularly pertained with protection of forest. All the features of entomophilous flowers except flowers with aroma, versatile stamens, pollen grain with sticky surface. Flowers provide reward to pollinators in the form of nectar and pollen grains. So when you say versatile stamens, that should be because entomophilous means the one that is getting pollinated by the insects. That is what with entomo. So flowers with aroma, yes, naturally when you have a good smell, the insects will come near it. It should have a sticky surface, yes. It should give reward in the form of nectar and pollen grains. All this is true. But what is that? The versatile stamen. What are those versatile stamen when you see? They are characteristic of anemophily, particularly that is getting pollinated through wind. So wind pollinated flowers are usually well above the ground. The stamens are exerted and often you call them as versatile anthers, so that they get easily dispersed. So that is with the versatile stamens. Secondary sewage treatment is mainly a physical process, a mechanical process, chemical process, or it is a biological process. Yes students, yes, you are all giving the right answer, very good. So what is with the secondary sewage treatment? It is particularly what type of process? Yes, it's a biological process. See, when you see the sewage pollution, particularly the primary treatment, secondary treatment and tertiary treatment we have. So what is there with the primary treatment? That shredding, flotation, screening, sedimentation you have with the primary treatment. What about with the secondary treatment? Here the organic matter, whatever is coming here, it is going to get decomposed by the microorganism. And those sewage fungus, by passing through that thing, what will happen? It will help in activated sludge method. So aeration helps in oxidation of the sludge. So the tertiary method is a physico-chemical treatment. So the correct one with the secondary sewage is what? You are making it act upon by the microorganism. So it is purely a biological process. So that you need to know. Whereas the primary one will come under the physical, where you do the shredding, churning, flotation, okay, all that. So it's more of physical come as a mechanical process. In which contraceptive method the semen will be without sperm? Condom, withdrawal coitus, foams, vasectomy. definitely during the intercourse the semen will be ejaculated. So in which of that the semen will not be there? So usage of condom or withdrawal coitus or forms, definitely there will be presence of semen except the vasectomy. Vasectomy, it is a permanent contraceptive method done in males, where there will be sterilization by preventing the transfer of sperm. So they will just cut the vas deferens and they just tie it up, so that the semen will not, the sperm or the semen will not reach into the penile organ. So that is with the vasectomy. Okay. The total number of teeth which comes only once in a lifespan, 28, 32 and 12. We know the milk teeth is 20 and the permanent teeth is 32. So 20 teeth of a child, they get replaced in the adult, right? We have in the human adult the 32. But the rest, what happens of this 20, the 12 teeth, it will never be replaced. They say in some of the theories, they say they are the one which becomes the molars and premolars. So they grow once when you go into a man life. So the 20 milk teeth of a child gets replaced in the adulthood by 20 permanent teeth. So but the human adult has got 32 teeth, the rest 12 can never be replaced if broken, because they grow only once in a man's life. So the answer is what? 12. Excellent. Very good, students, you are giving the right answer. So when synapsis is complete all along the chromosome, the cell is said to have entered a stage called zygotine, pachytene, diplotin and dikinesis. So which is the right option here? So students, when you look into it, when I say zygotine, it is the stage where it is marked by the beginning of the lateral pairing of the homologous chromosome at the chromosomal tip, and there is formation of that somatonemal complex which forms there. What about in the pachytene stage? There is complete lateral pairing of homologous chromosome along the entire length of the chromosome. So the DNA gets exchanged between the sister chromatids, non-sister chromatids. But what about with the diplotin? The diplotin is marked by breaking of the synaptonemal complexes, which is holding together those synapses, which in turn causes the separation of those chromosomes, except the point of crossing over. And dikinesis is the one which is marked by the separation of the homologous chromosome, where the nuclear membrane will also disintegrate. It marks the end of it. So when synapsis is complete all along the chromosome, the cell is said to have entered a stage of what? It is entering a stage of pachytene. So pachytene is marking the complete, where the synapsis is complete along the entire length. There is completion of lateral pairing of the chromosomes along the entire length of the chromosome and the crossing over. So what we call it as the pachytene stage. Which structure of the lung is directly involved in oxygen carbon dioxide exchange between the air and the blood capillary? Bronchi, trachea, alveoli, and secondary bronchi. So which of the stage is that? So we all know that directly the exchange is going to take place only within the alveoli. So you can see the alveoli where we see the pulmonary capillaries. So they are in direct contact. We have what is called the respiratory membrane, which is a part of the alveoli as well as the pulmonary capillaries. So the direct exchange between the oxygen and the carbon dioxide will take place only in the alveoli. So that should be the right answer. So you have nearly around some 6 layers that forms what is called the respiratory membrane. So you have a layer of surfactant, we will have the alveolar epithelium, and then we will have the the basement membrane, then we will have the capillary endothelium. So like that they will be arranged sequentially, where it allows the easy exchange of the respiratory gases. The correct sequence of cytochromes in ETC is site A, B, C, A3, cytochrome B, C1C, A3, cytochrome B, A3C, cytochrome B, C, A3A. So in electron transport chain, when you see, the ATP is produced by oxidation and reduction reaction in the mitochondria, where there is transfer of electrons, that is from one protein complex to another. So the electrons are actually transported from NAD to flavonoprotein and then to the cytochrome Q. So from cytochrome Q, what happens? The electrons will be transported to the cytochrome B, then C, A and then A3. So the right option is what? It is A3. So cytochromes are what? They are nothing but proteins which are characteristic for the strong absorption of that visible light, because they have that iron containing heme prosthetic groups. The mitochondria, which I said already, they have the 3 classes of cytochromes which are designated as A, B and C. Okay, so you can distinguish by differences in their light absorption spectra. The cytochrome of type A and B and some type of C, they are integral proteins of inner mitochondrial membrane. So how will be the sequence? As I already said, the electrons are transported from that NAD to the cytochrome Q. From the cytochrome Q to cytochrome B, then C, then A and then A3. So that is the right answer. Yes students, very good. You have given the right answer. Very good. You're doing a wonderful participation in these sessions. Excellent. So I hope this session would have been very interesting, come interactive also. So see you all soon in the next part 2 session of this mock 4 test questions. Okay. So thank you students.